research seminar. So a little bit about Emily here. So she came to our school she did her undergrad degree at Bryn Mawr College, got a Bachelor of Arts in Biology, and she obviously had quite a love for aquatic organisms because she did her undergraduate thesis on sturgeons, and she spent a few weeks on an offshore research vessel studying green crabs. And I'd like to think that that love of fish and aquatic organisms brought her here to our school. And she was brought on to help us with a National Science Foundation funded research project, which has been a large collaboration between Michigan State University and Archbold Biological Station here. And on top of that, she has also been working on an independent research project, which she has done a lot of work optimizing some new methods, which she's going to be telling you about today. I just want to say before she gets started that we wish you all the best in your future endeavors, and applications to grad school, and so on and so on. And with that, I will turn it over to Jones.
Hello, yes, my name is Lee Jones. In my project, you're hot and you're cold, eating cold tolerance of mosquitofish and freshwater habitats. So to begin with, we're going to talk about climate change. So while global temperature averages are increasing, there's evidence that climate variability is also increasing. There was an observational study by Cohen that found that winter temperatures have increased and severe winter weather events have increased in eastern North America and eastern Eurasia. However, warming temperatures and increased precipitation can also have a cooling effect on certain ecosystems. For example, warming and drying in the Sonora Desert in the southwestern U.S. has caused annual rains to come later in the year, which has resulted in colder winter temperatures. So freshwater systems like ponds and lakes are especially vulnerable to changes in climate variability. Ponds in particular, due to their smaller size, are more vulnerable to temperature variability as they don't have the depth or size to buffer against it compared to other larger ecosystems such as lakes. Increases in precipitation and evaporation also rapidly change the water volume, which can lead to larger swings in temperatures in those small ponds. So aquatic ectotherms are animals that are dependent on the external water temperature regulation, obviously on that water temperature, for most of their physiological and behavioral traits. It has even been described as the master abiotic factor for fish. So this means that long-term heat or cold stress can negatively impact fish survival in ways. It can reduce swim performance, can result in almost equilibrium, can delay or prolong growth, and can even worsen outcomes. So since ectotherms are so vulnerable to changes in temperature, we need to respond. We need to understand how they respond to increased climate variability in various. So the big questions that I focused on for my project was, do heat and cold tolerance ectotherms differ between freshwater systems of different sizes? And then does that range of thermal tolerance also change between those freshwater systems? And then I also looked at whether or not thermal tolerance changes over seasons as well, from late to fall, as that's when I was putting my sample. So to look at thermal tolerance in aquatic ectotherms, I chose eastern mosquitofish as my sample species. So eastern mosquitofish are a small freshwater fish native to Florida. They have been observed to have a large thermal tolerance range with robust body of study for their heat and cold tolerance. So there have been natural populations that have been found in waters with temperatures up to 39 degrees Celsius, which is about 102 degrees Fahrenheit, as well as waters that reach below 0 degrees Celsius, so 32 degrees Fahrenheit. So these species can obviously adapt to a very wide range of temperature extremes. They can also be used as an indicator species to learn about the biotic and abiotic state of the environment as they prey on small invertebrates and then also prey to larger freshwater fish. And then I also chose these two different freshwater habitats on or near Archbold Station. I looked at 2 scrub ponds and then 2 larger lakes, sampling these sites across 10 weeks through the end of summer and into fall, including the end of the wet season, to understand how depth and size influences thermal tolerance. So the 2 scrub ponds that I looked at were an old cattle pond and then a single. And these are permanent scrub ponds, so they do not dry out during dry season, like compared to like other scrub ponds do. These both have a depth of about 2 meters and a very small surface area. And then the two lakes that I looked at were Lake Annie here on Archibald property, and then Lake Espoga, which is located north in Highlands County. So Lake Annie is a sinkhole lake that is unusually deep compared to other lakes in the area. So it has a depth of about 20 meters. And then Lake Espoga is much shallower with a maximum of 3 meters, but it has a much larger surface area. And then the sample temperatures that are listed here for each of these sites is the temperature that I recorded when I was out there sampling. So here are some maps of my sites. So you can kind of get an idea of different size variation. So on the left, we have the 2 scrub ponds in the Lake Archibald property. Here are the 2 scrub ponds, and then here is Lake Annie. And then on this map to the right, you can see Lake Espoga with Lake Annie right down here precisely. So you can see that Lake Espoga is much, much larger. And then both of these lakes are much larger than the scrub ponds, which their surface area is highlighted in pink. So you can see how very small they are compared to that. So my predictions that I was looking at. So I predicted that because small ponds are more vulnerable to temperature changes, mosquito fish from these ponds should likely experience more temperature variability compared to the mosquito fish from the lakes, as the lakes should be more resilient to extreme temperature fluctuations due to their size. So I predicted that the mosquito fish from the ponds would have a higher heat tolerance and cold tolerance compared to lakes. So they should be able to tolerate higher heats and lower colds or colder colds. And then because of that, I also predicted that mosquito fish from the pond would also have a wider range of thermal tolerance compared to mosquito fish from the lake. And then my final question, I predicted that the thermal tolerance in mosquito fish would change over the seasons. This is because aquatic ectotherms, such as fish, can change in various physiological ways to adapt to changes in their environment. For instance, their heat tolerance can increase during the summer months when it's hotter and then decrease in the winter months when it's colder as a way to conserve energy. So I would expect that as the temperature falls from summer to fall, that tolerance for heat would lower while their tolerance for cold would rise. So for my sampling, I caught 24 juvenile mosquito fish each site twice, for a total of 192 fish from August to October. And here is a scheme of my sampling, so you can see which sites I sampled from which days. Some studies have found that there is a difference in tolerance between the different sexes in adult mosquito fish, which is why I chose juveniles to kind of avoid that confounding factor. So once the fish were caught, I acclimated in the lab for 2 days before testing. So I would sample on Monday and then run my trials on Wednesday. And during that time, the fish were not fed, so that way there would not be any energy expenditure or digestion during my trials. To measure the heat and cold tolerance, I ran trials to measure the critical thermal maximum and the critical thermal minimum of these fish, the CTmax and the CTmn. This is the temperature that fish would typically lose equilibrium at, which is the point where they can't right themselves and would die if left at that temperature. So it's basically a pseudo-lethal way of measuring their heat and cold tolerance. So this was my setup for the CTmax trials. So this cooler would be filled with water, and then these 2 heating coils would heat the water at an even rate of about 0.4 degrees Celsius per minute. So then once the fish lost equilibrium, they would be removed from there and placed in the recovery tank. And then after they were all removed and then covered, euthanize them and measure it to get their mass. And then for the CTmn trial, the procedure was basically the same, but the setup was pretty different. Using methods modified from Carnegie, I built this cooling setup and optimized it for mosquito fish, which really hadn't been done before. So it was a little bit of a struggle getting that to work. So I had these 2 different coolers set up next to each other. And in this larger cooler, I had these copper tubing that connected through these tubing to this cooler to a pump and a PVC pipe. So water would flow from this cooler through this pump, through all this copper tubing, and then out through this PVC pipe back to this. And this cooler would be filled with water, and I would add ice to it at 5-minute intervals to get a cooling rate of about 0.4 to 0.5 degrees Celsius per minute. And then same as with the CTmax trials, once the fish lost equilibrium, they would be removed and then processed. So my main variables of interest that I looked at were temperature, which was the CTmax temperature, my date of sampling, and then location, which include all 4 sites, cattle, sinkhole, lake, and this book. And then I ran a generalized linear model with CTmax or CTmn temperatures as a response variable, and date of sampling and location as my predictor. So here is the heat tolerance by sampling date using the CTmax temperatures, which range from 41 degrees Celsius to green, plotted on the y-axis, and then the dates from August 3rd to October 12th here on the x-axis. And then I have these different colors represent the different sites. So red is the cattle pond, green is, red is the sinkhole, green is the cattle pond, teal here is Lake Annie, and purple is the sinkhole. And then I also included a line from when Hurricane Ian hit, as that most likely caused a change to the water volume and change to the temperature as well. So according to my model, I found that sampling date was significantly associated with heat tolerance. As you can kind of see here, the CTmax is gradually decreasing as time goes on for the different sites. And then here is the cold tolerance by sampling data using the CTmn temperatures, ranging from about 5 degrees to 11 degrees, plotted across the same dates, the same colors, and the same model for hurricane. So according to my model, I also found that sampling date was also significantly associated with cold tolerance. So while there is not visually as extreme of an effect, there is still an effect as CTmn does decrease as aka they were able to tolerate colder temperatures as the seasons went on. So the significant association between both the CTmax and the CTmn sampling date did support my prediction that thermal tolerance did change. And so next up, we have the heat and cold tolerance by sample location. So here are the CTmax temperatures plotted here on the left in red, and then the CTmn plotted here right in blue. So we have the temperatures plotted on the y-axis with a range of about 1 to 3 for the CTmax, and then about 5 to 11 for the CTmn, and then the location plotted here on the x-axis. So according to my model, I found that location was significantly associated with heat and cold tolerance. As you can kind of see here, the scrub ponds did have a higher average CTmax compared to lakes, and a slightly lower CTmn average compared to the lakes. So this does support my prediction that the mosquito fish from the ponds had a higher heat tolerance as well as a cold tolerance compared to mosquito fish from the lakes. This also means that the mosquito fish from the scrub ponds had a wider range compared to those from the lakes. And here is another graph that kind of shows that a little bit better, as it shows the difference between the average CTmax and the average CTmn for each of the 4 locations. So the difference between the CTmax and CTmn is plotted here on the y-axis with range of 41 to 43 here on this top part, and then about 7 to 9 on this bottom part. I will note that this is on a pretty small scale if you look on the y-axis, but even small temperature or degree changes in temperature can have significant impacts on ectotherms. So this does also support prediction that mosquito fish in ponds had a wider thermal tolerance compared to the mosquito fish. So in summary, to answer my 3 big questions, does heat and cold tolerance ectotherms differ between the freshwater sizes? Yes, I found the mosquito fish from ponds had a higher heat tolerance and had a lower cold tolerance compared to mosquito fish from the lake. So they were able to tolerate higher heats and colder colds compared to those from the lakes. And then does the range of thermal tolerance change? Yes, I found that the mosquito fish from the pond had a wider thermal tolerance range compared to mosquito fish from the lakes. And then does thermal tolerance change over seasons? Yes, as time passed, both heat and cold tolerance decrease. And these results are actually pretty interesting because temperature responses at the hot and cold end are not actually often associated with most ectotherm thermal physiological studies. So this is actually pretty cool that I found. And that's actually about it. So some acknowledgements. I'd like to thank Mr. Judson and Vincent Rockamount for all their support and guidance. Without them, this project would not have been possible. haven't even known where to begin without just with help with the CTmax and CTmn stuff, as well as all the stats. That was a lot of work. And I'd also like to thank the Herb Lab, Rachel, Teresa, and Cammie, as well as the previous interns, research assistants, making the work environment so supportive. I'd also like to thank Sarah Fitzpatrick at Herb Lab, as they're the ones running the study that I'm working on, and their funding from the NSF is what funded my internship. So it's the whole reason why I got to be here. I'd also like to thank the Archibald staff for maintaining the station and making such a welcoming environment, as well as my fellow interns and friends here at Archibald, and as well as my family and friends back home, for their just general support during my time here. So, are there any questions?
I'll ask maybe the obvious question, is it genetic or if you swap the fish from one to the other with the chain?
That is a really good question, because it is kind of hard to like tease out those different factors. The project that me and Jessica are working on is actually kind of trying to get at that question. They're running a very long-term mesoconism experiment by subjecting different mosquito fish to different temperature ranges. That they're going to do genetic studies at the end and see if there is indeed a change. Hopefully here that isn't too much of an issue, since these are separated populations. There might be some plasticity in the thermal response, but since all those different habitats would have a kind of similar plasticity response, they both should be able to like somewhat change to the environment. But since they're in those habitats long term, there should also be some adaptation. But whether or not it's actually genetic, I can't really answer without looking at the genetics. But since they're separate populations, that should be a confounding factor, I guess. That's answers your question.
Yeah, I thought that was really cool. I was curious, you know anything about dispersal between those populations? I imagine there are some so much flooding here, disperse and
Yeah.
Big ones.
Yeah, and that usually does happen with the little ponds, but since these are permanent ponds, while there might be some dispersal out, as long as the base population is still there in the pond, they're not really, they don't like move and come back, because usually once they disperse, then the ponds dry up, kind of like stuck in wherever they disperse to. So there's not really like much movement to it from populations. Both the sinkhole and the cow pond are permanent populations. See that there.
I was wondering if you took any more platform.
There was some phenotypical variation in the mass and length, which I did look at the statistics on that, but since it wasn't really relevant to my project, I didn't really include it here. There did seem to be, as time passed, some of the populations were having smaller sizes, but that might have been due to like disease outbreaks in certain areas. Like the cattle pond that we sampled from was definitely having some type of issue. We're not really sure what it was, but that might have been a factor. I did save some of my samples that I collected for Sarah, because she's running the genetics from the mesocosm experiment. I did save some samples for her because she wanted to look at the genetics of it. So whatever results she get will be kind of cool to look at. But other than that,
Can you explain again why you chose juveniles and with this kind of the
yeah, so the reason I chose juveniles. So there have been other heat tolerance experiments that have been run that found that there was a difference between male and females mosquito fish. It might have been due to the size of the mosquito fish, because the females tend to be much larger than the males. But I didn't want that to be like a confounding factor, so I used the juveniles since they haven't matured yet, and also should be around the similar size. So that's basically why I did it, was because you can run an experiment like this with the two different sexes. You just have to account for that confounding. It kind of makes the statistics a little more complicated. So I just didn't really want to have to deal with that. So I just looked at the yes.
Oh, awesome job, Emily. I had a school how you built all these different like games and stuff. I was just wondering if you could talk more about your process for figuring that out. So, how many failures that you may have taken?
Yeah, so again, I was using methods from this paper called alternative to cooling as well. There are like kind of like heating things that exist, or like the alternative, the opposite of like heating ones where there are like cooling pumps that can cool the water. They tend to be pretty expensive. Like the ones that I found were all like hundreds, if not thousands of dollars, or were also just like not being produced anymore. So I was using this method where they were using this copper tubing in a separate tank that connected to the tank that they were testing the fish in, and using that ice to cool the water, copper tubing at like an even rate. So if you just add the ice into the water directly, it can make the temperature kind of go wonky where it like will spike up much higher than you want it to. But yeah, it was a little hard, especially since I didn't really know the exact minimum temperature that the fish would like get down to. So I didn't know if I'd like water down to like below 0 or not. Thankfully, I didn't have having to do that because that would be a really complicated thing where I would have to add like, I would have to like turn the pumps and I had heated water back into it and then add salt. It would have been a whole thing, but thankfully I didn't have to do that. But I did have to run various trials to kind of get the methods and the optimization kind of down to make sure that it would lower at an even rate and like how much ice I would have to add. So yeah, there were several weeks where I was just running tests through this, which does take a bit of time. It can take about like 40 minutes for just the water to drop down because it was at a pretty slow rate. But I was able to get a pretty even rate of about 0.0, which I was pretty proud of because that was a little hard to get.
There's a couple questions online. So what was the most challenging part and your favorite part?
Yeah, so the most challenging part would have definitely been this critical thermal minimum, especially doing the trials because I had to basically remove any like factors of like some fish being in the lab longer than others and like kind of trying to remove as many confounding variables as possible. I basically had to run all the trials in one day, which did take a lot of time. So very long days running all of our trials at once. So that was definitely hard, but it was definitely worth it for the data. But then the favorite part, I don't know, I really like going out and just sampling mosquitoes. That was a ton of fun. You just kind of sit there, you just catch them with your little net, or well, we had a bigger net, but yeah. You know, just have to like look at them. And at Lake Annie, there's this, I forgot to include the picture of it here, but there's this special morph of mosquito fish at Lake Annie, the Dalmatian one, which have these really cool black and white spots on them. I actually managed to catch a few while I was sampling. I was like, oh, that's cool. I didn't include them in my sampling because I didn't want to remove them from the population, and I also didn't know because they only affect males. So I didn't include any of them, but it was pretty cool to catch them.
Another question, do you have a favorite sampling site, which one and why?
Lake Annie was probably the best just because it's such a nice location. It's also really easy to sample there because there's a dock that's like right there by the entrance, and mosquito fish like to just kind of swim around in the shallows of that dock. So you can basically just like step out into like the 2 feet deep water and just stand there. Mosquito fish just kind of swim around. So it's really easy to sample there. That was like the one site I never had like any difficulties finding the amount of fish that I needed. So that one was definitely.
With your heating cooling coolers, you think you're going down and get the results like mini coolers that have cool heat stuff better than just like regular coolers?
I mean, these were insulated coolers. You can kind of see that these were both Coleman coolers, so they are insulated. So hopefully that didn't have an effect. This cooler here is also the one that just puts their heat tolerance experiments. So I basically just got a larger version of that. They are insulated coolers, so wouldn't have any troubles with that.
Mentioned some climate change questions.
talk about. Explain a bit more about
Yeah, so the main kind of point with the climate change is there's a lot of research done on the general like heating of climate change and like the effects of hot temperatures that will have on various different ecosystems. But the climate variability itself is something that's not actually worked on as much. There's not as much research done into like what effect like having more extreme temperatures might do. There's also a lot of like conflicting research and observations on whether or not it is an impact of climate change or if it's just like a natural variation that's happening as time goes on. It's really kind of hard to prove that. But there is some data that suggests that there is variability that is increasing, as well as like certain extreme weather events like hurricanes and flooding and stuff like that, which can also like have kind of immediate impacts on like heat and cold. Especially with like these ponds, just increased precipitation, but also water ones have kind of like an inverse impact on the actual temperature in the pond itself. Yeah, for future questions, the main thing I kind of want to focus on is this thermal tolerance range, because that will kind of indicate how well certain ectotherms could adapt to those like extreme temperatures, especially since there is some data that suggests that maintaining a wider thermal tolerance is actually much more costly than maintaining one for a stable one. So there is like some evidence that like maintaining the ability to adapt to such wide temperature extremes is not as good as being able to adapt in one narrow range. So learning how like different species can adapt to that and also the effects that might have on them. Lots of ways you can move forward with this. So that's kind of like the main thing I want to do. That kind of ability to adapt to that range of extremes. Any other questions? Yeah. Yeah, there are actually several specific genes that they've looked at in heat tolerance. I won't be able to name the specific ones because they have names and there's like a dozen of them. But since temperature does impact so many different traits in fish, so it's not just like just dissolved oxygen, it's like almost everything that a fish can regulate is regulated by temperature in some way. So there's a lot of different genes that could impact that, but there is, since some fish are like plastic in the way that they can adapt to changing environments, and so they can kind of like regulate their ability to tolerate certain temperatures, like on a more smaller time scale than just like evolution. There is probably some like genes or certain pathways that regulate their ability to do that. So which ones exactly those are, I couldn't say. But I do know that there has been a lot of study which genetics impact which heat environments, which is what Jessica and Sarah are working on, trying to see like which genetics change with heat tolerance.
Thanks everybody for watching. I hope you can hear everything.